Hey everyone, welcome back to Cells to Cell. This episode slash video is going to be kind of fun and entertaining. Nick and I are actually going to ask each other quizzes, trivia about regenerative medicine and stem cells in general. So we've got 3 quizzes. It's going to be entertaining. It's going to be a bit interesting to see how much we know about our own field.
Yeah, especially a first year versus a second year. So I'm clearly already at a disadvantage, but that's besides the point.
Okay.
So we have a couple quizzes we can choose from. I think we should just do the 25 question quiz today.
Okay.
I think that'll take enough time because I'm sure as we ask questions, we'll kind of elaborate and kind of explain. And we're actually probably going to put a link to this quiz in the description, or these quizzes in the description. So if you want to, test your knowledge a little bit before you watch us and have all the spoilers, go ahead and pause the video, go play those. It probably shouldn't take too long for someone to just play it, maybe by themselves or with somebody, and work together. But then come back and just see how we do, and any question we might get the same questions wrong. We might be able to elaborate a little bit. So how was the first question?
Oh, you asked the first question.
All right. So, question number 1. Pluripotent stem cells, or iPS cells, are reprogrammed adult cells produced in the lab to take on characteristics of which type of cell? Oh no, I'm not going to do very well on this. Is it A, somatic stem cells, B, tissue-specific stem cells, B or C, embryonic stem cells, or D, mesenchymal stem cells?
I actually know this because I'm a second year and he's a first year and he hasn't got that lecture yet. But it's C, embryonic stem cells.
Yeah, I knew that.
You knew that?
Yeah, and I can explain why. Wait.
So the guy that discovered iPSCs is such an interesting story. His last name is Yamanaka, and because he discovered it, he had these proteins named after him. But basically, when all the cell surface markers and the proteins that exist on an embryonic stem cell, this PhD student named Yamanaka decided to put all those same proteins on an adult stem cell, so a stem cell that's already differentiated. And what happens is that that stem cell actually went back in time and behaved like an embryonic stem cell, which is interesting. So those group of proteins, all in combination that he put in an adult stem cell, they're called Yamanaka factors. And these Yamanaka factors made adult stem cell look like an embryonic stem cell. And the reason why this is so groundbreaking is because adult stem cells don't really differentiate that well into all different kinds of stem cells. And when we say pluripotent, pluri means plural, so like a lot of different things. And when you take an adult stem cell, turn it back into a pluripotent stem cell, now you can take this stem cell and turn it into something else. For example, I can take a fat cell, give it Yamanaka factors to make it embryonic. So then from this stage, then I could turn it into bone. Because naturally, it won't really go from fat, and I can't put any proteins and things to make it a bone. I'm going to take this fat cell, put some Yamanaka factors to make it induced pluripotent stem cells. So iPSC, you're inducing them to be pluripotent. And pluripotent usually is affiliated and associated with embryonic stem cells. That's why it's so controversial to use embryonic stem cells, but embryonic stem cells are really good for their stem-like properties. But take this fat cell, turn it into an induced pluripotent stem cell, which then it can turn into all different kinds of cells: fat, cartilage, bone. So yeah.
Hold on, there's been an issue. Okay, well, if we know the answer, it doesn't matter.
I have the answers. We'll go.
Okay, perfect. I was just trying to get it to where we can actually click and take the quiz. So, okay.
Well, here we can just boom.
It doesn't give it. You have to click on it.
Hold on. Show answers. Well, we'll do that.
Okay. Yep.
Embryonic stem cells.
Didn't see it.
Question 2.
All right, let's do it.
The first reports of successful mammal cloning date to the A, 18th century, B, 19th century, C, 20th century, or D, 21st century?
I'm gonna go with C because
20th
the
century?
20th century.
So like 1900s?
Yes, because I'm pretty sure wasn't it was Dolly the sheep. She was like the 90s, I believe, or the 80s. They were actually able to do it. They were actually take full complements of one Dolly's DNA, put it into like an empty cell or an empty embryo, and they were able to actually create a clone of Dolly the sheep. So they had two Dollys. But the clone actually did not have the normal lifespan because of genetic complications, which is probably assumed to happen when you take a full complement of genes out of one cell and put it into another. So
that is right, 20th century. Okay,
yes.
So the first successful mammal cloning was actually not too long ago.
No, they've been working on it for a really long time too.
Interesting.
But most of that stuff, I don't even know how they would have done it in the 18th and 19th century because of genetics. Although you had Mendelian genetics, dating way back, there was still no way to like guarantee a copy. So, all right, question 3. Regenerative medicine relies heavily on the use of.
Oh, come on.
Do you just want to answer it?
Stem cells.
Yes.
What do you mean?
It's probably going to be stem cells. Let's check.
Is that the
I would
answer?
assume so. Yep, it's
All
stem
right.
cells. So that's, we've been covering that for the last, I don't know how many episodes. But yeah, okay. I think I saw that this has like a 67% like answering the question right rate.
Okay.
So
hopefully we can do better.
Hopefully we can do better
being
with
students.
our scientific backgrounds and being students of this field.
All right, question number 4. Adult stem cells are to be harvested and transplanted into a patient. Where would these cells most likely be found? Interesting.
It's going to be tough.
Yeah, it's very open-ended, but let's see. A, in an embryo. B, in muscle tissue. C, in human bone marrow. Or D, in a clone.
I'm going to have to go with bone marrow.
Bone marrow, why is that?
Because that's where, now you're making me second guess myself.
No, I'm just asking.
So bone marrow has a lot of dividing and proliferating cells. And what was adult stem cells, right? So when we talk about adult stem cells, we're talking about mesenchymal or mesenchymal stem cells, or I immediately think of hematopoietic stem cells, or HSCs, as well as there are other multipotent and unipotent stem cells that can differentiate into one or a couple different cell lines. But when I hear bone marrow, I hear stem cells.
That's actually true because it says where would these cells most likely be found?
Yes.
And we're talking about adult stem cells, they're mostly in the bone marrow.
Mm-hmm.
And that is correct.
Yeah. So and maybe some people thought embryonic stem cells, and usually we don't usually touch those.
Yeah, we
especially taking them from an adult. That is, we could talk about multiple episodes of the ethics and morals of stem cells in general, but especially embryonic stem cells. There's a lot of public outcry about using those, even in studies from cell lines established years ago.
Yeah.
So, all right, let's get into question number
5.
Number 5?
Yeah.
All right. Scientists implant genetic information from one species into another to create a customized strain of mice for research. What are these scientists creating? A, transgenic microorganisms. B, transgenic animals. C, genetic mutations. Or D, pharmaceutical modifications.
Okay, they're not transgenic mutations, they're not pharmaceutical stuff.
Mm-mm.
So it's either A or B. Can I see what they were?
Yeah.
Transgenic microorganisms or transgenic animals? I'm going to say transgenic animals. I'm going to go with B.
Especially when we're talking about mice.
Yeah.
When I think of microorganisms, I think bacteria.
Bacteria.
And that kind of stuff, or archaea. So, let's see.
Let's see if that's
Transgenic
right.
animals.
That's true.
That is
Transgenic
true.
animal. Okay, 100% so far.
So, a lot of testing of stem cells, go through what are called animal models before, where you test with transgenic animals, normally mice or rats, where you can actually kind of see how they work. Because believe it or not, humans are very closely related to mice and rats, even though they might be infiltrating your house. So, all right,
Okay.
next question.
Question number 6. In regenerative medicine, once the biomaterial matrix is developed, the scaffolds must be able to A. Leave, B. Made smaller, C. Dissolve, or D. Remain unchanged. So once a biomaterial matrix is developed, the scaffolds must be able to leave, be made smaller, dissolve, or remain unchanged.
So, my initial gut reaction is dissolved, but I have no knowledge yet of scaffolding.
Yeah.
So the way I'm thinking about it is, if I were to administer an implantation, let's say of a tendon, and I have stem cells on basically a scaffold that would mimic the size and shape of your tendon, and I would implant it, put it on, allow the cells to grow. So instead of waiting for attachment, like the cells are growing. That's why if you were an athlete, you would sit out for a while, or if you were just kind of like a light person, you would just rest. But did it say it was biocompatible?
Biomaterial matrix is developed.
Yeah, so it's probably biocompatible. I'm gonna go with dissolve. I'm probably wrong, but
if I think about it, I honestly would go with either with dissolve or remain unchanged.
Right. I was kind of between one of those two, but again, like if it's dissolved,
then it would lose its right. So like it might.
So I'm gonna change my answer to remain unchanged.
Okay.
Let's just kind of see.
Let's see.
I'm probably wrong.
Oh, you were right. It's
dissolve. I should have stuck with my gut.
No, I'm gonna give you that point.
Oh, I should have stuck with my gut. No,
I'm gonna give you that point.
No, it's okay. I should have stuck with my gut.
So okay. In the future podcast episodes, we'll go over.
We'll talk about biomaterials and matrices and matrices and all that kind of stuff, and
for sure.
How you can maybe utilize, and how we struggle to grow 3D cultures and everything like that, which is 3D culturing is something that I have found a new interest in.
Yeah.
As well. So, all right, that was 6. Let's go to question number 7. Adult stem cells are undifferentiated cells that are also known as A. Somatic stem cells, B. Embryonic stem cells, C. Pluripotent stem cells, or D. Hematopoietic stem cells. You're getting layups, by the way.
A.
Somatic
stem cells.
Yes.
Somatic just means body.
And that is correct.
Yay. Embryonic and pluripotent are kind of one of the same. When you have embryonic stem cells, they are pluripotent. Hematopoietic stem cells, those are just like the certain kind of stem cells that turn into your blood products.
But those can be differentiated into different types of blood cells. So whether it's red blood cells or red blood corpuscles, my anatomy and physiology teacher from undergrad would enjoy me saying that instead of red blood cells. But it could be
corpuscles?
Yeah.
I've never heard that.
They don't have a nucleus.
Oh.
They're not nucleated.
Oh, okay.
Yeah, they lose their nucleus as they differentiate.
Interesting. Okay.
So they're not cells, they're corpuscles. They just carry hemoglobin.
So
which is cool, which is super cool. But that's besides the point.
What's interesting is that adults do have hematopoietic stem cells.
Correct. Yeah.
But it's just like this is an open-ended.
But the question asks, adult stem cells are undifferentiated cells. So hematopoietic stem cells are not undifferentiated.
Are they
not? Can change. They can.
Okay.
They can change.
Yeah. Right.
Somatic stem cells.
They can't really change. Right.
So okay.
Question 8. Somatic stem cells are undifferentiated cells that are also called, embryonic, adult stem cells, totipotent, or zygote stem cells.
Say it again. You went through it pretty quick.
Somatic stem cells are
Mhm.
undifferentiated cells that are also called embryonic stem cells, adult stem cells, totipotent stem cells, or zygote stem cells.
Adult stem cells.
That was like
That-
one of
okay.
the same questions. I'll give you another
Yeah.
one. I'll give you another one.
Okay. All right.
Was that right though? Yeah, that was right. Oh shit, my bad.
Oh.
We're gonna get demonetized.
Sorry.
We're gonna download a bleep thing.
Reproductive cloning is also called somatic cell nuclear transfer, adult stem cell transfer, nucleic acid recombination, or protein synthesis. Ooh.
Say A through C again.
Reproductive cloning is also called somatic cell nuclear transfer,
Mm-hmm.
adult stem cell transfer,
Okay.
nucleic acid recombination, or protein synthesis.
Nucleic acid recombination. It's
Re-
either A or C.
Reproductive cloning.
I'm between A and C. I'm gonna go with. I'm gonna go with C because the word somatic in A is really throwing me off because when we talk about if we're talking about a zygote is
DNA.
when sperm meets egg,
Yeah.
right? So that is an embryo. So if you're doing zygote transplantation or whatever, those are absolutely in no way, shape, or form somatic cells. So I'm gonna go with C.
No.
Ah,
So the answer is
Really?
A. Reproductive cloning is also called somatic cell nuclear transfer. I guess nuclear transfer.
yeah.
I don't really know. That's
That's
a
a
tough,
tough
one.
one.
That is a tough
I'll have
one.
to look that up. I'll have to. All right.
Question 10.
I got one wrong. It's okay. Implanting a gene from one organism into an embryo will create A, xenograph, B, transgenic animal, C, new species, or D, a mutant.
What's the question?
Implanting a gene from one organism into an embryo will create a xenograph, transgenic animal, new species, or mutant.
I've got to see the options. I would say transgenic animal. I'm not really sure what a xenograph is. A new species.
No.
Maybe. I mean, maybe. A mutant.
If it's just implanting one gene, it would not create a new species.
But into an embryo, so it affects the development of the whole.
Yeah, okay, maybe.
But I'm gonna say transgenic animal. Who knows? Yeah.
It is a transgenic
Transgenic animal.
All right, 11.
One benefit of creating a genetically modified mammal is to. So, one benefit, okay, of creating a genetically modified animal is to produce animal models for research, create a clone of an organism, produce a zygote, or harvest embryonic stem cells.
I'm going to go with A.
Produce animal models for research, I would say
Yeah.
the same.
Yeah.
Because that's a benefit of
'Cause
genetically modifying.
Correct, and when scientists work with like animal models, they either add a gene to try to add a protein or something that they can monitor, and that might affect a drug, or they'll do what's called a knockout, and they'll take a gene out, so that you can compare it from a healthy animal to a knockout animal that is missing something.
Yep, that's right.
Yep.
Nice job.
All right, 12. When creating a cloned mammal, what structure contains the genetic information the scientist intends to clone? A blastocyte, an enucleated oocyte, or
Oocyte.
is it an oocyte?
I think it's-
We called it an oocyte because
oocyte.
we're from the south. Somatic cell or surrogate mother.
I would say then a nucleated oocyte or a blastocyte, but I'm gonna go with the oocyte.
Okay.
No.
Ooh, it's
what?
a somatic cell. All right.
When creating a cloned mammal, what structure contains the genetic information the scientist intends to clone? Oh wow. Oh wait. Oh.
What contains the genetic information?
Okay, so you can
So
clone
it's
something
where?
literally from any body cell, I guess.
Yeah.
That's fascinating.
Okay.
Okay, got one wrong.
101 wrong. We both have one wrong.
When researchers apply for a patent from the U.S. Patent and Trademark Office, for what process are they seeking approval? Creating a transgenic animal, reproductive cloning, therapeutic cloning, or nanotechnology?
Nanotechnology.
You think so?
That's such a weird question.
That is such a weird question. Let's see.
I don't think. Can you patent? No.
Creating a transgenic animal.
I didn't know you had
Wow.
to, I didn't know you could patent animals. I didn't know you could patent the animals you test. You learn something new every day.
Oh wow.
That's pretty cool.
That is interesting.
The only reason I said nanotechnology is because of
That's something.
it just sounds like something you would want to patent for.
Yeah.
But I guess it doesn't really matter because people patent drugs all the time. So, all right. All right. Which stem cells are most often used to avoid rejection by the patient's immune system? So, what is most often rejected, or what is most often used to avoid rejection? Okay. So, adult stem cells, therapeutic stem cells, synthetic stem cells, or cloned stem cells.
I'm gonna go with B, therapeutic stem cells. I really hope that's right.
It's adult
stem cells?
So-
What are you talking about?
So the way that therapeutic stem cells make sense, and you could probably call them therapeutic stem cells. But when you take adult stem cells out, you're taking out MSCs,
Yeah.
and MSCs are not only mesenchymal stem cells, but they're also medical signaling cells. And they actually go to the site and help with, graft versus host disease, inflammation, and that kind of stuff by changing the microenvironment. So I can kind of see where you'd get confused, but those MSCs are really, really important. So it's okay. Hey, both got you
we're
wrong.
bombing this quiz.
it's okay.
Okay.
It's all right.
Which structure has the potential to develop into any other cell within the body? Zygote, embryoblast, totipotent cell, or ovum?
Oh man, I did not take developmental biology. This is tough. But
No,
I'm
no,
gonna
it's
go
not.
with I'm gonna go with I'm gonna go with B. I'm gonna go with an embryoblast.
Really? Come on.
Well, like a like when I guess
I would
it's a
say
zygote.
no. I would say totipotent, right? Because that's one step above pluripotent. Totipotent means it can make any total.
Oh yeah.
They
And
didn't
I
say
guess,
the word stem, but
I guess. Okay. With your assistance, I will say totipotent cell because that does make sense. But a zygote is totipotent.
yes.
So, an embryoblast is totipotent.
Yes.
So,
I mean,
yeah,
they
it's
really
totipotent.
got us with the jargon here.
Yeah.
But I know what you were trying to say.
Yes, you have to really read into the question.
Yeah.
Okay. Replace, reduce, and refine are alternative scientific methods researchers use to examine cell tissues, maintain all lab equipment, design experiments that do not use animals, or request grants for special funding. Replace, reduce, refine are alternative scientific methods researchers use to what, Heem?
This may be crazy, but I want to say almost the lab equipment.
Replace, reduce, refine. Yeah.
Replace, reduce, refine.
Because you'd want to put new technology in that could help reduce the amount of technology that you're using, so you can reduce waste and stuff, and then
Refine.
continuously refine how you do that. But
Or design experiments that do not use animals. So it's either B or C.
yeah. So what are you gonna pick?
I'm gonna go C. Design experiments that do not use animals.
I like that.
Yes.
I like that.
There we go.
All right.
Because maintain does not really mean replace, reduce, refine.
Right.
Maintain means you keep what's there. Okay. The first step to building a new organ using regenerative medicine is to build a platform, foundation, scaffold, or basement. Scaffold.
Yeah. Yeah, we had that question earlier. It just gives it a basis to grow on, basically. So yeah, I'm gonna go scaffold.
Scaffold is a word used
All the
in
time.
this context.
Yep.
Okay.
Boom.
18.
All right, 18. What animal was successfully cloned in a research facility in Scotland in 1996 from an adult stem cell? Oh wow, I can't believe I gave you this answer.
Sheep.
Yeah, it was a sheep. Sheep is a D. Yeah, Dolly the sheep. There we
You
go.
are the quiz.
I am the. Hey, like I said, cellular molecular biology, man. I guess I learned something.
Okay. What approval process do biomedical researchers seek when they create a transgenic animal? Submit documentation to the Department of Agriculture. Register the process with the Internal Revenue Service. Apply for a patent from the U.S. Patent and Trademark Office, or petition the National Institutes of Health.
They want to get a patent.
I think so, because that was a question earlier. That's true. You apply for a patent.
Which I think it's crazy. I feel like I did hear that somewhere in like undergrad that people patent like the mice lines that they use, but I didn't realize that like everybody tried to do it.
So if you create a mouse lineage that
Mhm.
has diabetes because you genetically engineered it, you could patent that.
I guess.
You could patent a diabetic
And
mouse.
you could probably just keep producing more mice and sell them off to people studying diabetes.
This industry is wild.
Yeah, there's
Wild.
all right. Anyways, adult stem cells are to be harvested and transplanted into a patient. Where would these cells most likely be found? So adult stem cells to be transplanted. Where would they most likely be found?
Human bone marrow.
Well, let me read them the stuff. It would be in an embryo, in muscle tissue, in human bone marrow, or in a clone. And Heem said in bone marrow.
Once again, because
Once
it's
again.
talking about adult stems.
Yep, in bone marrow, like we said earlier, the hematopoietic stem cells and the mesenchymal/mesenchymal stem cells
In adults are found
in adults are found mostly in the bone marrow.
Yeah.
Well, ones you can harvest easy
Yes.
enough.
Okay. 21. A group of similar cells from the same origin performing a specific function. Oh my god, you mentioned this in the podcast. Okay, so a group of similar cells from the same origin performing a specific function. Is that a tissue? Is that an organ? Is it a muscle? Or is it a stem cell?
Hold on, read the question one more time. I don't want to sound like I don't know it, but I want to hear the question one more time.
A group of similar cells
Okay, cells. It's a tissue.
from same origin.
It's a tissue.
It's a tissue. That's right. An organ is
An organ is made of tissues.
an organ is made of different tissues. Or
But
similar.
the same tissues perform, or different tissues performing the same function.
Yeah.
It's okay. All right, 22, right? Yep.
Yes.
Surrounds and supports the cells that form tissues and organs in the body. It is created and maintained by cells. Is it A, a scaffold, B, a biomatrix, C, stem cells, or D, extracellular matrix?
C, stem cells. It surrounds and supports the cells from tissues and organs in the body. Yeah, because stem cells are found as a parasite. They're right next to every single tissue type, every single organ type. Oh no, what?
It's the ECM or the extracellular matrix. So I was thinking about this, like,
That's so wrong. Oh my
you
god.
think that's wrong or you're wrong?
No, I think that's so bad that I'm wrong.
I mean, let's take bone for example, because cells in your bones, to make them hard, like on the outside, osteoblasts and osteoclasts, they make an extracellular matrix called hydroxyapatite, and it's like got calcium and magnesium in it, and that's why calcium's important in our diet. One of the reasons calcium's important in our diet and magnesium's in our diet. But they actually break it down, and like there are like little cells, like right, and then they just have like this hard matrix around them, and that's why it gives them their stiffness and their rigidity, and why it's so hard to break them. So
See, when it said surrounds and supports the cells that form tissues and organs,
and we're taking a regenerative medicine.
I
quiz.
thought stem cells. I
Yeah.
really thought stem cells, but
It's okay.
extracellular matrix. Okay, 23. The heart is an example of a tissue, gland, bone, or organ.
It's an organ.
All right. I'm gonna give you one more.
Oh, I thought I was gonna struggle on that one.
24. Where are human embryonic stem cells derived from? Bone marrow, zygote, scaffolding, or somatic tissue? Human embryonic stem
I'm
cells.
gonna go with the zygote. I would say so.
I would say so as well. Nice.
Yeah.
Okay.
All right, you want to give me the last question, or yeah, you can have it. You can have it.
This is an easy one.
I think I won. We'll watch it back. I'm kidding. It's okay. All right, last question of the video. So blood stem cells are also known as somatic stem cells, totipotent stem cells, hematopoietic stem cells, or embryonic stem cells.
See, hematopoietic stem cells.
All right. So, all right. Well, I mean, a couple of the questions. I think we both got 4 wrong, or we got 4 wrong combined. I think 4 or 5 wrong.
4 out of 25.
4 out of 25. So what is that like?
Times 4. That means
What?
16.
No.
100 minus 16 is 84. And 84%.
Ooh,
Higher than the average, right?
higher than the average, but you know, I feel like it was maybe a little bit worded different. But again, this was a great quiz, a great constructive quiz because it was like, you know, the basics. Like it kind of laid out everything. So, do you have anything else?
No, but
No? I think
we have
that
a
was
couple
fun.
extra quizzes to take later on. That's future videos, but
Yeah.
hope you enjoyed this. We're trying something new, and
Yeah. Hey, thanks for watching the Cells to Cell podcast. We'll see you later.